Now, if the B cells are moving, even within the germinal center, from the light zone to the dark zone, remember, that also means they have to keep changing their chemokine expression because that's what's allowing them to move. Otherwise, they'd be tethered in one place or the other. So they are constantly changing what's on their surface to allow them to move. And again, germinal centers last for about 3 to 4 weeks, and then they start to resolve, and the mature B cells, the product of that, either become memory B cells or plasma B cells. Here is a picture of what a germinal center looks like under the microscope. And again, you have a lighter zone here because there's fewer cells, hence the name, and the dark zone is where the cells are really tightly packed. This staining picture shows you where the different cells are. T cells are in blue, and you find them in the T cell zone outside the germinal center, but also in the light zone. The red cells are those follicular dendritic cells that have antigen. And the green cells are mostly B cells, but this marker here is actually showing you dividing cells. And again, this is to indicate that most of the B cell division is occurring in the dark zone. But you can see here interaction of some dividing B cells, the follicular dendritic cells, and the T cells all in the light zone. Now, B cells that cycle between light and dark are again undergoing affinity maturation. So they mutate in the dark zone, but they have to go back to the light zone to encounter their antigen again, because what if the mutation means that they no longer bind their antigen? So this process is cyclical, and the cells go back and forth, and some cells survive this process, but a lot of the B cells will not survive this process because the mutation will not help them bind their antigen any better. And again, once the process is done, then the germinal center goes away, gets small, and eventually disappears, and those mature B cells go back to the bone marrow as memory B cells and plasma B cells. And plasma cells can also go throughout the body. Okay, so let's get to what affinity maturation actually is. Germinal center B cells undergo V region, so the variable region created by VDJ recombination that you learned about, somatic hypermutation. So this is not about any gene rearrangements, it's about specific point mutations within that already rearranged gene. So you have a primary antibody over on the left that has no mutations, and then you start to see some mutations in the variable region. The other thing that happens through this process is a class switch to a different constant region, such as going from IgM to IgG. This is just a graph showing you about how many mutations can occur in a germinal center. And by day 7, you can have a few, and they are localized in certain regions within the variable regions. And by day 14 and day 21, you have more and more mutations. Remember, we're selecting for only the mutations that can still bind antigen. And that's the point of the B cells constantly going back to those follicular dendritic cells to say, okay, can I still bind antigen well? And if they can, then they'll present that antigen again to more helper follicular cells and get even more help to proliferate and divide. And this is just a picture of that. I've got B cells, there's 2 different B cells here, an orange B cell and a yellow B cell. Now this one, its mutation has actually increased its affinity for antigen, but the cell in yellow has actually lost affinity. So what happens in the light zone is that this high affinity antibody on a B cell is pulling antigen off, then it's going to interact with the T cell receptor on those follicular helper T cells, and this low affinity antibody can't do anything anymore, can't bind to the antigen anymore, can't present more antigen to get help from T cells. So this B cell is a failure and actually will go on to die because it no longer binds its antigen. And again, this is just the same process shown here and about how this B cell is getting help and this B cell is no longer getting help. And the B cells that receive the most help go on to re-enter the dark zone, and they go through a whole nother round of mutation, and the process continues for several rounds. And the goal is to select for antibodies that have a higher and higher and higher affinity for that one antigen, and to eliminate the cells that don't. Now, how does this mutation actually occur? That's where we bring in our AID enzyme. Now, Cytidine Deaminase actually takes the base C in DNA and makes it a U. And normally you don't have any U's in DNA, you only have uracils or U's in RNA. This enzyme only works when the DNA is single-stranded. Now remember, most of the time our DNA is never single-stranded in our cells. It's double-stranded. However, if a gene is being transcribed, the two DNA strands have to open up so that RNA can be made off of one of them. And that's what provides the window of opportunity for AID to come in and mutate a C in a single-stranded piece of DNA. And that's what's shown here. This is showing you how it actually attacks the base. And then you end up with a U in the DNA instead of a C. The cell recognizes that as a mutation, but it has a few different ways that it might handle that mutation. It might simply take the uridine and convert it back to a base. That is mismatch repair, and what normally gets inserted then is an A or a T, but that's still no longer a C, so you get one base pair mutation in your DNA. The other possibility is that the cell actually removes that base because it views it as damaged. Then you don't have any base there, and the cell actually has to stick in a new base. And when it does that, it seems to preferentially choose C's or G's. So with the same initial mutation, you can end up with different ways of repairing that mutation. The other thing that occurs, in particular when you go to no base, when the uridine is removed, is you end up with a nick in that one strand of DNA. The two sides are no longer attached when the base is removed. That then engages another repair enzyme to fix the nick. And a lot of species, other than humans, do something called gene conversion, but what humans have evolved to do is called class switch, and that's how we're going to get our different isotype and classes of antibodies. So this again is just the initial process of mismatch and base excision in the DNA. The AID comes in, mutates a C on a portion of the DNA that's single-stranded, and then the cell can actually remove that base. But this again is the only way that you're going to get class switching. You don't get class switching if it simply repairs that U. So, mismatch repair and base excision repair pathways are how you're going to get this somatic hypermutation. And the hope is that, or what germinal centers are based on, is that some mutations increase your affinity of your antibody for your antigen, and some of those mutations decrease it. But at least some of them increase it, otherwise the whole process wouldn't work. Now, as I said, AID also initiates class switching. That starts with still the same mutation of a C to a U, but then the U gets removed and you have a nick in the DNA. That opens up the door to allow the cell to consider switching classes. So if you see here, you've got your VDJ region, and maybe you have some mutations in there that have increased affinity. But now that gene has to link with one of the constant region genes. You've got M, you've got the different IgGs, you've got A and E as different antibody classes over here. So there still needs to be some DNA rearrangement at this point in order to get a C region linked to the variable region. And we're going to break down this process a little bit more on the subsequent slides. So first, before each, actually I'm going to go back one slide just to point out to you that you've got these S regions before each gene. They're repeat regions, but they also contain a promoter that promotes transcription of each of those genes. And that's going to be important for this process. Now, a cell will actually loop out the DNA in between the variable region, or the VDJ, and all of the classes or isotype genes it doesn't want to get to the one it does. So this cell is trying to make an IgE, for instance. So it's going to get rid of all of the Gs. The S regions can bind to each other, and you actually then can get excised from the DNA. Now, this is an irreversible DNA recombination event. So once a cell has isotype or class switched, for instance, to IgE, it can never go back and make IgG because those genes are lost from the genome. It could potentially, though, go on in another round, perhaps, to make an IgA because that gene is still there. So what you end up with is S regions that are both repeat regions and promoters in front of the constant region that you want for your antibody. Now, the next thing that happens is that, remember, AID made that initial mutation because the gene was being transcribed. So the DNA recombination is actually occurring almost simultaneously, really, with transcription. So the RNA is transcribed, and then those S regions, because they have repeats in them, can bind to one another, and they form this quadruplex of just loops bound around that can interact with each other. This then is going to get excised out of the RNA, not out of the DNA. And that's just shown in a close-up here. And basically, the S regions are more or less an intron, and they get spliced out of the RNA as an intron. Now, how do you get all the different subclasses of antibody? What determines that? It actually comes from the help from the follicular helper T cells, because cytokines that the T cells make turn on those different promoters in front of each of those constant regions and allows the cell to choose which type of antibody it wants to make. That's going to be directed based on the type of antigen and the experience the T cell have in seeing that antigen, not only presented by B cells, but also presented by innate cells even earlier. So you can see that IL-4 can inhibit certain classes and promote others, and that's true for all of the cytokines. They promote making some types of antibody, and they actually inhibit making others. And if you think about when you learned about helper T cells, we talked about type 1 and type 2. They make different cytokines, and then you have the result of that is that different classes of antibodies get made. So to summarize T-dependent antibody production, B cells first have to interact with and acquire their antigen, present it on MHC class II, and get help from a T cell. The B cells will actually induce a more generic or regular helper T cell to become very specifically a follicular helper T cell, which is best suited to really help a B cell make a lot of antibody. The TFHs provide several different signals to B cells. They help them survive, they help them proliferate, and they help them differentiate eventually into those plasma cells and memory B cells, which is the positive outcome we want from a germinal center formation. And remember, in that germinal center, the goal is to increase the affinity of the antibodies being made by the B cell. Because the stronger the antibody can bind, the more antigen it's going to bind, and the better job it's going to do of trying to eliminate that pathogen. All of these mutations actually then require a lot of DNA repair enzymes. So there's a lot of DNA repair going on in a maturing B cell in a germinal center. Now, as I said at the beginning, some antigens don't require T cell help. Some antigens can stimulate B cells, and there's no T cell involved. There are actually two different kinds of T-independent antigens, just called 1 and 2, and this table is a comparison of different types of antigens. Now, antibody response in infants. You can get antibodies very early after a baby is born if they are challenged with some infection to a T-dependent antigen. You can also get it to a T-independent type 1, but not type 2. I'll come back to that. If you have no T cells, you don't make antibodies to T-dependent antigens because those B cells require T cell help, and they'll never get activated and they'll never make antibody. But if you don't need T cell help, then you can get antibodies to the different T-independent 1 and 2 antigens. Of course, it's really only the B cells that respond to T-dependent antigens that actually prime T cells, because these B cells don't ever interact with T cells, so they're not going to prime them. Polyclonal B cell activation. Now, we normally think of B cells having one specificity and binding to one antigen, and that's true. But with a T-independent type 1 antigen, it gets activated not based on the B cell receptor, and I'll show you a picture of that. So actually you get B cells with a lot of different specificities activated, only for T-independent type 1 antigen. For T-dependent and T-independent type 2, it actually is antigen specific. In particular, T-independent type 2 have repeated epitopes. And this last part of the table here just shows you examples of some pathogens that have these different types of antigens. This picture, I just want to drive home the point that T-dependent antigens, the first signal is coming from the antigen. The second signal is always coming from direct contact with a T cell. With a T-independent antigen, whether it's 1 or 2, there is no T cell, and the signals are coming to the B cells themselves. But you still need 2 signals. Example of a T-independent type 1 antigen. Again, imagine that this is a TLR receptor on a B cell, and that you have a pathogen here that's expressing whatever that PAMP is. Although these B cells have different antibody specificities, they all have the same TLRs on them, so they're all going to be activated by the same PAMPs. When you have a lot of that antigen around, you'll basically activate all of these B cells regardless of their B cell receptor. And so you have all these different colors here showing you that you've got polyclonal activation of B cells that is not antigen specific. At low levels of a T-independent type 1 antigen, you might get a specific antibody response. But this is very rare, and that's the most common scenario. Now that's different than a T-independent type 2 antigen. Again, they still don't require T cell help, but this is a very antigen-specific response. And you have your surface IgM here, your B cell receptor, and multiple receptors are being engaged simultaneously by the same antigen. So it's a polyvalent antigen. It's often something like a polysaccharide that has repeated epitopes of the same thing, but it could be protein as well. That's enough to stimulate this B cell to make antibody, although it does seem to get some survival signals from the BAF cytokine made by, for instance, a follicular dendritic cell. But it doesn't require any cell-to-cell contact. Now, marginal zone B cells, which are found mostly in the spleen, in humans, but also found in some lymph nodes, and in other animals like mice, only found in the spleen. Their job really is to respond quickly to antigens found in the blood. And they're very good at responding to T-independent type 2 antigens. However, when you're first born, you don't have any marginal zone B cells. And you start to develop them when you get to be a few years of age, and it's not really until after you're 10 years old that you have a full complement of marginal zone B cells. So one of the reasons that babies and little kids can get so sick is because they're missing this component of their immune system. And in particular, they don't have the ability to make antibodies to T-independent type 2 antigens. And adults do have that ability. We'll come back to that in the vaccine lecture, and I'll give you an example of how this matters for vaccine development. Now, once you get B cells making antibody, you have them distributed and functioning in different places. Now, different classes work in different places. For instance, IgA works a lot in the gut. So that means there has to be transport mechanisms to get IgA in particular into the gut. Neonatal Fc receptor ensures that IgG gets into a developing fetus so that fetus can benefit from its mom's memory B cell responses. IgA and IgG antibodies do go around in their different locations, and of course their job is to find a pathogen or a toxin and neutralize it. And we've got different receptors here, and you'll learn more about complement as well. But when an antibody binds to whatever its antigen is, you don't just want that floating around in your body, you need to have a way of eliminating it. And complement and Fc receptors facilitate that. So I just want to briefly talk about some of the different characteristics of the different classes of antibody, but you'll learn more about this in the lectures on immune responses to different pathogens. A lot of the antibodies can neutralize. They can simply bind to a pathogen and kind of neutralize it. Some of them opsonize and then help a phagocyte, for instance, eat that antigen better. Some of the subtypes of antibodies actually help other cells to work to kill pathogens. And then again, some of them can activate complement. Where are the antibodies found? Again, mostly IgA is what can cross epithelial layers, and that's how it can get into mucosal sites. A different subset of antibodies cross the placenta to get into a fetus. Almost all antibodies can move between within tissues, so extravagate into all tissues. Of course, most of our infections aren't in the blood, so antibody needs to get either to the skin or to the lung or to a site of infection. And this just shows you the levels of antibody found in the serum. Here's a table just to give you an example of, and this is bacterial antigens,